Hello, everyone. So welcome to another ATBC webinar series. I would like to thank Patricia and Zach and Juan and Natalia for organizing this really great activity for the Neotropical chapter. This is a new activity for ATBC, and I'm really thrilled to be able to participate. I'm also thrilled for having been selected as executive director of the ATBC. And since I'm just starting my term and a lot of people still don't know me, I thought I would take a couple minutes to introduce myself. I'm originally from Brazil. I was born and raised in São Paulo. And here you can see me in this picture together with my cousins. I'm around 5 years old and I'm holding my little vase with cacti here, which I used to take with me everywhere I went. And also, as a child, I was also a stamp collector, but I actually used to only collect stamp of flowers and plants, which has always been my big passion. When I was 16, I decided I wanted to be a botanist. And so I went on and got my undergraduate degree at the University of São Paulo in Brazil. And during my undergraduate, I also conducted a lot of field work in Amazonia, as I was part of a team that was working on a field guide for the Duque Reserve in central Amazonia, close to Manaus. After my undergraduate, I then moved to St. Louis, where I got my master's and my PhD degrees at the University of Missouri, St. Louis, and also a postdoc at the CCSD, the Center for Conservation and Sustainable Development of the Missouri Botanical Garden. And after finishing my postdoc, I then moved back to São Paulo, where I got a position as a professor at USP, the same institution where I had gotten my undergraduate degree. And this where I have been working since 2003, although for the past 2 years, I have been based at Berkeley, where I have a 2-year position as a visiting professor. I'm now at the end of my term here at Berkeley and getting ready to move to Brazil now in July. So as you can see here, I feel that I have really been connected with nature and plants as long as I can remember, really. And so it's perhaps not really surprisingly that a lot of the research that is now being conducted in my lab focuses on understanding the assembly and evolution of biotas. This is a question that has really intrigued ecologists and biogeographers for a very long time. And for the Neotropical region in particular, a lot of the high species diversity that is found in this region has been attributed to a lot of geological events, such as the Neogene tectonics, or the Miocene marine incursions, Pliocene river drainage formation, or quaternary climatic changes. Although we also know that ecological aspects such as pollination, seed dispersal, herbivory, insect-plant interactions, among others, also represent really key drivers of species diversity in the neotropics. Despite all of these hypotheses, we still don't know the exact factors that have actually shaped the neotropical biota, which really calls for a truly integrative and multidisciplinary studies. Ideally involving a variety of taxa, although before we can actually do cross-taxonomic analyses, we actually need to have detailed information on the ecology, evolution, systematics of the various organisms, which unfortunately is still not available for all neotropical organisms. And so before we can actually conduct these cross-taxonomic analyses, we still need to conduct in-depth analyses of individual lineages. The question is, which organisms to select? So given that we cannot study all organisms at once, which organisms should we select as our models? So I don't know what you see when you look at a neotropical forest, but I see pink flowers, well, yellow flowers and pink flowers and lianas and more lianas. And coincidentally, these are all members of the plant family Bignonieae, which is the group that I have selected as my model for studies in the neotropics. I am not the first person to select the Bignonieae as a model for studies in the neotropics, and Al Gentry, one of the most important botanists of all times, who really made key contributions to our understanding of the history of neotropical forests, also selected the Bignonieae as his model group. And in fact, a lot of the hypotheses that he proposed about the origin of the neotropical flora were actually based on his studies and observations of the Bignonieae. One of the reasons why the Bignonieae is such a special plant family is because it's a key component of tropical forests and especially diverse in the neotropics. The family has a reasonable size, around 850 species of trees, shrubs, and lianas that occur in all major ecological zones and are extremely diverse morphologically and ecologically. So what I thought I would do today is to first tell you a little bit about the in-depth studies that we have been doing with this key neotropical clade. And then I'll tell you a little bit about some of the cross-taxonomic analyses that we have been conducting in the Amazon and the Atlantic forest. And then I'll finalize talking about some of the implications of this research for science outreach, education, and also biodiversity conservation. So let's start with our in-depth studies of the Bignonieae, starting with systematics, which is really the foundation of any biological work. In my lab, we have a deep appreciation for natural history, and we conduct a lot of field work, a lot of herbarium work, and this is really important as this provides the basis for the preparation of species lists for many different parts of the world, which ultimately led to the preparation of a world checklist, which is really critical for anyone doing work on the Bignonieae. We also have a phylogeny for the whole family. This phylogeny has identified 8 main different clades in the family, one of which is the tribe Bignonieae, which has actually been the focus of most of the research in my lab during the past years. The tribe Bignonieae includes around half of the species in the family and actually represents one of the most diverse and abundant clades of lianas in several neotropical ecosystems. Lianas have key roles in forest regeneration, on ecosystem level functioning, and they also account for up to 40% of the wood stem density and diversity of several tropical ecosystems. One of our first steps for our work with Bignonieae was to build a phylogeny for the group, which actually then served as basis to establish a new generic classification for this group. And this was really a critical step of research because it allowed us to identify meaningful units for subsequent work. But even though this phylogenetic framework was really crucial and represented an excellent starting point, this initial phylogeny only had 1/4 of all species recognized in the tribe. And so it was really clear that we needed species level phylogenies and detailed monographs of all 400 species in the tribe if we really wanted to have a full understanding of the diversification history of this group. I was very lucky to have encountered a series of very talented students who were also interested in this group and who decided to take on individual lineages for in-depth phylogenetic and monographic work in this group. So they essentially, each student picked a clade to work on based on the suitability of that group to address particular evolutionary and ecological questions that they were particularly interested in. And through this amazing collaborative work, we were then able to cover every single clade in this group and to gather very detailed knowledge on all 400 species of Bignonieae. In general, the work of these students included a phylogenetic and biogeographical component. Each one of these students also encountered new species during their field work. and they also produced a taxonomic revision of their group of interest. All the studies used a standardized morphological matrix and also a standardized georeference locality database. And so by gathering all the data in this standardized fashion, we were able to build very large and comprehensive database for all the species in this group. And this then represented the basis for a series of other evolutionary and ecological studies in this group, which takes us to the second component of our studies with the Bignonieae. Since the tribe Bignonieae represents one of the most abundant and diverse clades of lianas in neotropical forests, we really wanted to understand why these lianas have been so successful. It had been hypothesized in the literature that their success was due to two major traits. The presence of a specialized wood anatomy and also the presence of tendrils. When we looked at the wood anatomy, we found that in these lianas, the cambium becomes discontinuous in equidistant portions, leading to the formation of phloem wedges. There is variation in these wedges, with this trait becoming more and more complex over evolutionary time. So in other words, the ancestral condition in the tribe is the presence of 4 phloem arcs, here depicted in black, from which 4 phloem wedges subsequently evolved, here depicted in red. And then we see the evolution of multiples of 4 phloem wedges in blue and multiple dissected phloem wedges in green. And it's very interesting that each of these traits actually only evolved a single time, showing that the wood anatomy is actually a very conserved and sort of hard to evolve trait. So this might actually help us explain why this trait is not found in any other angiosperm family. When we looked at the wood anatomy in further detail, we found that a lot of the finer scale differences, so at the cell level between the regular and the variant phloem, increased over time. So this is probably what allowed these plants to become more and more efficient in terms of their phloem conduction and storage capacity over time, which is another factor that might help us to explain why these plants are so successful nowadays in neotropical forests. When we looked at tendril evolution, we found a very different pattern. Namely, we found that tendrils are quite labile, so they're not as conserved as the wood anatomy. And in addition, we found that tendrils were originally triphid, from which we see the evolution of simple tendrils multiple times, and then going back to triphid, and we also see the evolution of multifid tendrils. And this is actually the opposite of what had been hypothesized by Darwin in his book, Climbing Plants, as he initially thought that the evolution of tendrils would have taken place from simple tendrils towards more complex features. And so this really showed that the story was much more complex than we initially anticipated, which might actually have been due to the fact that tendril morphology is associated with very few genes that can be turned on and off quite easily. The extrafloral nectaries were another trait that we were particularly interested in. In Bignonieae, these extrafloral nectaries are actually modified trichomes with very diverse morphology. And it turns out that these nectaries evolved multiple times after transitions from forests to savannas, suggesting that these traits might actually represent adaptations to the savanna environments. We then wanted to get a better understanding about the function of these nectaries, but actually, counter to our expectations, we actually did not find any evidence of a protective role. So this result really led us to, we couldn't understand why, and this led to a second study with a much more broader sampling of individuals and locations. And in this broader study, we actually found that the abundance of extrafloral nectaries was quite variable among populations. So while 4 populations, the ones depicted here in red, included a much higher number of extrafloral nectaries that were visited by large and aggressive ants such as Camponotus and Pseudomyrmex, the other 6 populations here depicted in black were actually had much lower number of extrafloral nectaries and were visited by much smaller ants such as Crematogaster and Cephalotes, both of which did not provide any kind of protection to those plants. These results are perfectly consistent with the geographic mosaic theory of coevolution, which hypothesizes that populations of a single species actually undergo different selection pressures and likely evolve different features in different geographical areas. So these findings actually helped us to understand why we did not find a protective role in our initial study, and which turned out to be due to the fact that we had only sampled cold spots. So this result really highlights the importance of scale in ecological studies. Finally, we were also interested in studying the evolution of flower morphology and their respective pollination systems. We first analyzed a series of discrete traits, and we found, first of all, that most floral traits are very homoplacious and essentially change a lot from one lineage to the other. We also found that traits that are associated with the same pollination system generally evolved in a correlated fashion. And finally, we found that there are actually multiple ways to produce flowers associated with individual pollination systems. So in some instances, we see punctual evolution with direct transitions between different pollination systems and flower morphologies. And in other cases, we actually see gradual evolution, which includes the evolution of intermediate and actually sort of maladapted flower morphologies between the different pollination systems. When we looked at the evolution of continuous traits, we found something very interesting. We found that the traits associated with the external flower whorls, so calyx and corolla, are phylogenetically conserved. So in other words, calyx and corolla show similar sizes within individual clades. On the other hand, the traits associated with the internal whorls, so the Androecium and Gioecium, they are actually very homoplastic, showing a lot of variation in size with individual clades. So this means that closely related species actually show corollas and calyces with sizes that are more similar than expected, but at the same time, the stamen styles and stigmas, they are much more variable than expected by chance, which is likely associated with the maintenance of reproductive isolation that we see among species of Bignonieae nowadays. Well, a good understanding of character evolution is really critical for us to determine the ecological forces that explain community assembly and phylogenetic structure. And so we then decided to use the morphological information that we had gathered and look at the effect of phylogeny, environment, and morphology on the assembly of Bignonieae communities. Our specific objective was to evaluate the role of environmental filtering and competition mediated by pollinators for Bignonieae community assembly. And we found 3 main things. So first of all, we found that communities of Bignonieae are not phylogenetically structured. So in other words, close relatives do not co-occur more or less than expected by chance. We also found that competition for pollinators is not an important driver of Bignonieae community assembly. And finally, we found that species distributions are actually driven by the environment, with habitat filtering likely representing a very key driver of community assembly in this group. So this then leads to the final component of our detailed studies with the Bignonieae, which is biogeography, which is essentially community assembly at broader scales. The first step of our study then was to time calibrate the phylogeny of Bignonieae. And what we found was that this plant group originated in eastern South America, where we currently have the Brazilian Atlantic rainforest, here depicted in green. And this happened in the Eocene at around 50 million years ago. This group then reached Amazonia, depicted in blue, where it diversified extensively, returned to the Atlantic forest multiple times, and also reached Central America, here depicted in yellow, several times, and then reached the dry areas of central Brazil at around 30 million years. But if we really want to understand the origin of the Neotropical biota, then we also need to put this study into a broader context and understand where these species came from. So this led us back to our broad phylogeny of the Bignonieae. Then we coded the distribution of all Bignonieae species using very broad biogeographical realms, and used our extensive fossil record to time calibrate the Bignonieae phylogeny. And what we found is that the Bignonieae actually originated in South America at around 56.8 million years ago, after the separation of South America and Africa. We have then 4 main lineages of the family that diversified in the Neotropics exclusively. So this is the Tribignonieae, the Tabebuia alliance, the Jacarandas, and Tourettii. And then one lineage, the Catalpi or Roxyliclade, reached North America and Asia through Beringia, and 2 other lineages reached the Old World, namely the Pantropical clade and the Paleotropical clade, which diversified extensively in continental Africa, in Madagascar, and also in Australasia. So once we reached this kind of detailed knowledge of the Bignonieae, we then started to wonder whether the patterns that were recovered with this plant family were exclusive to this family or could actually be generalizable to other groups. And this led us to venture into some cross-taxonomic analyses using other model groups for which similar kinds of detailed data were available. In our first attempt to conduct cross-taxonomic analyses to understand the assembly and evolution of the Neotropical biota focused on Amazonia, which, as we all know, represents one of the most biodiversity-rich biomes on Earth. This study had 4 main objectives, and the first of which was to characterize the spatial distribution of selected Amazonian taxa. And for this component, we selected some clades of plants, birds, primates, and butterflies as our focal taxa. And what we did here was to build large data sets and detailed distribution maps for all of our organisms of interest. After characterizing these organisms, we then moved on into building the phylogenetic history for each of the taxa selected using genomic data. And the third objective was then to characterize the geological and environmental history of Amazonia. And we were particularly interested in answering 3 main questions. The first of which was, what has been the history of environmental change across Amazonia from the late Neogene to present? The second was, when did the Amazon River drainage form? And the third was, what was the Amazonian landscape like before the Amazonian River formed, and how did it change after the river formation? The fourth objective of our study was then to combine all the data that we had gathered in objectives 1, 2, and 3 to then reconstruct the history of Amazonian taxa. Our first question was, so how old is the Amazonian biota? When we look at birds, we see that birds have been in South America for a very long time. Actually, since the late Cretaceous, even though major diversification of birds only really started in South America at around 65 million years. When we look at primates, we see that primates have also been in South America for a very long time, since the early Miocene. Same applies to butterflies, who have been in Amazonia since the Miocene. This is the same thing was found for plants who have been in Amazonia for a very long time. So in other words, our data is really consistent with the hypotheses that forests were established in South America in the Paleocene, but actually became more abundant in the Eocene. Now, so this part of the study allowed us to determine that how old was the Amazonian biota. In other words, how old have different organisms been in Amazonia, including fossil organisms. Now, in order to understand how old is the modern Amazonian diversity, we actually needed to look at the age of the living taxa and the terminal tips of our phylogenies. So starting with birds, when we look at the ages of the living taxa, we see that a lot of these species only go back to the Pleistocene or to the Pliocene. And this goes on with many different lineages of birds. The same applies to primates. If we look at capuchin monkeys, we see that the living species are also quite young. Same thing applies to squirrel monkeys. Same applies to butterflies. And the actual living species of butterflies are very young, with most of the butterfly divergencies having actually happened in the Pleistocene. And even though some lineages of plants are slightly older, a lot of the living species that are currently in the Amazon also go back to the Pleistocene, Pliocene boundary. And we see the same pattern with many different lineages. So after having determined the age of the Amazonian biota and the living species, we wanted to get a better idea of the drivers, what led to the diversification of Amazonian species. And when we look at this example from trumpeters, which actually became nowadays a really nice textbook example about Amazonian diversification, we see that a lot of the clades are separated by rivers. So in other words, this study suggests that rivers have acted as a really important barrier for bird diversification. And the same pattern is observed with primates. And so we have clades that are restricted to the right bank and to the left bank. And a lot of the primate sister taxa being separated across the Rio Negro and the Rio Branco, and having diverged only less than 2.5 million years. And even when we look at plants, so initially we didn't think that rivers would represent barriers for plants, but we conducted a very detailed study sampling populations of many different species in 20 sites over 50 kilometers. And we found that while the Rio Branco is not a barrier for plants, so you can see here in this graph in green, the L and R, and you see that L and Rs are intermingled, showing that there's these populations from the left bank and the right bank are not isolated. But then when we go to the Rio Negro, which is a much older and broader river, we see that the populations from the left bank of the river and the right bank of the river are separated, suggesting that even for plants, river can indeed represent barriers for gene flow. So out of this study on the Amazon, we have a couple take-home messages. So first of all, we learned that the elements of the modern Amazonian biota have very old roots, going back to the Cretaceous-Paleogene boundary, with modern-like rainforests becoming more abundant in the Eocene. Despite that, we also see that a lot of the living vertebrate species originated during the Pleistocene, which is only around 5 million years ago, and the same applies to several plant species, although some living plant species are older than that. We also see that the origin of some components of the Amazonian biota are associated with the establishment of the river drainage formation, also suggesting that the formation of the river drainage was much younger than previously thought. But a full understanding of the assembly of the Amazonian biota still depends on a higher amount of phylogenetic studies and a deeper understanding of the past history of this extremely dynamic vegetation and river drainage formation. Our second study, cross-taxonomic study, focused in the Atlantic rainforest. And we actually, in this study, we wanted to take advantage of all the data that we had gathered for and what is the tempo and mode of biotic interchanges between Amazonia and the Atlantic forest. So if we go back just to remind us a little bit, so we know that forests, we learned from our previous study that forests seem to have been established in South America in the Paleocene, but only became more abundant in the Eocene. And we also know that there was a trend of cooler temperatures and drier seasons during the Oligocene. And when we look at our data, we also see that, for example, our Bignonieae lineages occupied drier environments at around 30 million years. So this is consistent with the hypothesis that the dry diagonal that currently separates the Amazonian and the Atlantic forest formed at around 30 million years. And when we look at data from other clades, we see here, for example, we have in green an Amazonian clade and in pink a predominant Atlantic forest clade, and we see that these lineages diverged at around 30 million years, which is again consistent with the starting to form the dry diagonal during the Oligocene, following the trend of cooler temperatures and drier seasons that were found at the time. Despite that, we still see some connections between taxa in the southern portions of South America. And so it seems that the dry diagonal formed from north to south, with taxa, some taxa still being exchanged through southern corridors of forest during the Oligocene. We also have some botanical evidence that there was a northern connection between the Amazon and the Atlantic rainforest. And when we look at some of our taxa, we do see evidence, phylogenetic evidence, that there was a biotic exchange between Amazonia and the Atlantic forest through northern corridors during the last 5 million years. Suggesting that even though the dry diagonal started to form in the Oligocene, this dry diagonal was not fully stable, and there were corridors of forest that were formed at different times, allowing taxa to go from one biome to the other. And it's interesting that a recent study that looked at exactly the same question using very different data found patterns that were consistent to our observations. In fact, we see, for example, that a lot of the older bird splits are found in southern South America, corroborating the hypotheses of older forest corridors in southern South America, while a lot of the younger bird splits are in northern South America, corroborating the hypotheses of younger forest corridors connecting Amazonia and the Atlantic forest through the north. And we also know that throughout the Miocene, and in fact until today, we also have forest biotic exchanges that happened through gallery forests that have connected the Amazon and the Atlantic forest throughout time. So some of the take-home messages from these studies are that, first of all, so I think we improved substantially our understanding of the assembly and evolution of the Neotropical biota through these studies. Despite that, we still have a great need for additional in-depth and multidisciplinary research of individual lineages, as well as we also need additional comparative cross-taxonomic analyses and additional geological work if we want to really understand how the Neotropical biota was assembled. Now, why does this all matter? And I mean, as you could see from my studies with taxonomy, ecology, evolution, and biogeography, these are studies that are not immediately applied. So I have actually had people ask me, so why does this matter? And I mean, I feel that this really matters because this is very relevant for conservation. And I'm really convinced that academics really actually have the power to change the world. But in order to do that, we actually have to stop talking only to our peers. So I really like this quote from Savo Hileta from University in South Africa, in which he says that research and creative thinking can change the world. And this means that academics have enormous power. Despite that, academics are not shaping today's public debate. So instead, their work is largely sitting in academic journals that are read almost exclusively by their peers. And in fact, we see that up to 1.5 million peer-reviewed articles are published annually, but a lot of these are ignored even by the scientific community. In the humanities, for instance, 82% of the articles are not even cited once. And I feel that this really suggests that a lot of great thinking and many potentially world-altering ideas are not getting into the public domain. But the question then is, like, why then are the academics not doing more to share their work with the broader public? And Savo, in his paper, he mentions three main factors. First, that there is a narrow idea of what academics should or shouldn't do. Second, a lack of incentives from universities and governments. And third, a lack of training in the art of explaining complex concepts to the lay audience. I really feel that this is really a time for change and that the academics really need to start playing a more prominent role in society. And I love this other quote by Savo in which he says that quality academic research and innovation are crucial, but it's equally important to get ideas out into the world beyond academia. This could really make a real difference in people's lives. But the question then is, so how can we get others to listen, non-academics to listen to what we have to say? Well, most people are not experts in our fields, but they're really interested in learning more. And so I feel that we really should try to disseminate cool research findings in a way that everyone understands. We also need to talk more about the value of nature and the importance of conservation. And we really need to be creative and open-minded about the platforms that we use to share our work. So for example, blogs and popular articles, radio interviews, podcasts, TED Talks, movies, animations, TV shows, exhibits, mobile apps, citizen science, and children's books. So you name it, I feel like there's a lot of tools that we could use out there. We have been doing a lot of outreach articles. So we have been trying to explain details about our research to the lay public. And this has been very positive as we have been getting lots of questions and interested people trying to really understand what are we doing. We have also been involved in an alliance of world scientists that have sent out several warnings to humanity, calling for urgent and transformative changes to meet the goals for conserving and sustainably using nature. We have also put up some calls to action for even biologists and trying to get biologists to engage more in science, conservation, and policy. We have also been giving a series of radio interviews and have even produced some documentaries on our research on geogenomics, and also in some about our Amazon work. In this short documentary called Amazon Expedition, we essentially show to the general public all the work that we do. We also show them the beauty of Amazonia, the importance to preserve it. And in this trip that we did for field work, we actually also had documentarists and photographers and artists joining us on our boat during field work. And this was an extremely interesting and rich experience that actually led to an art science exhibit that we launched last year. And in this exhibit, we started with a historical module in which we talked about all the research that has been done in Amazonian since the 17th century. We then had a science module in which we talk about all the research that is currently being done in Amazonia. We then had an art module in which we showcase many different artists that have done fabulous work in Amazonia. And we even showcased the work of some of the artists that have joined us in different field trips. So here's, for example, the work of Juliana Albuquerque Leukovics. And in this work, she's a photographer who took lots of photos of animals and plants in the Amazon, printed them in leaves of Amazonian plants. And as the art exhibit developed, these images were disappearing, and it was an analogy to what is happening with the Amazon nowadays, which is really disappearing before our eyes. We had another work, for example, by Alberto Baraya, who is a Colombian artist. And Alberto has been doing his own herbarium or artificial plants. He travels around the globe and he compiles his herbaria. But these are all artificial plants. So in our exhibit, he put together this greenhouse. And when we look from outside, you're very keen on going in and seeing these plants. And once you realize that these are all artificial plants, it really brings you a very uncomfortable feeling, which actually is his idea is to show people that this is what will actually happen in the future if we don't take care of biodiversity. In the evenings, we showcased many different movies about the Amazon. We also had different workshops and activities showing people a little bit about the work that we do. And this also led to a series of popular articles calling the attention to Amazonia and to preserve the Amazonian rainforest. It also led to a documentary that is available on YouTube. And we had so many visitors, and the exhibit was so popular that we actually won an award for one of the 30 best art exhibits of 2018. Apart from the art exhibit, we've been engaging in different TV shows. For example, we participated in a show that was organized by the TV Globo. This is a very powerful TV station in Brazil. So imagine in Brazil, you have 180 million people, and this is a national TV that everybody watches. And this was essentially a show about the Bignonieae and this plant group that is so dear to us. And to our surprise, we had more than 5 million people watching this show and paying attention, wanting to learn and understanding the importance of this plant group. We have also been working on a mobile application to collect citizen science data for Bignonieae phenological research. Flowering of the Bignonieae is triggered by dry climate, by lower temperatures, and also increasing daylight. And because plant phenology is an important indicator of climate change, the information gathered through this app will actually allows us to better understand plant responses to climate change, while also engaging the general public into environmental issues. So essentially the way it works is that, so people just take photographs of their plant, and they submit through this app. And through the app, we're able to identify these plant species and give back, so the actual species identification, while also collecting the phenological data. We have also been working on art science books and focused on Amazonia, and also Amazonian coloring books, and inviting students to, from early age, to start to learn more about the Amazon. We have also produced articles on local newspaper devoted to kids. And here, for example, in this article, we suggested movies that they could see about the Amazon during their weekend. So essentially trying to get kids to learn more about the Amazon. And I also try to go to schools and read books about the Amazon and try to tell kids about the importance of the Amazon and biodiversity in general. And it's actually pretty remarkable what kids can achieve. This is actually an example from a project done in the class of my younger daughter. And the whole class, each student wrote a letter to Senator Feinstein. And they wrote this letter asking the senator to vote for the Green New Deal, and also showing their concerns about the environment and asking Senator to do something. And we were actually surprised that Senator Feinstein actually responded every single letter. And so getting kids engaged at a very early age can be extremely powerful and really makes them feel, well, act as citizen and already start to care and to do something about climate change and biodiversity conservation. So I really feel that we scientists need to be very active in science outreach, as this really is the way to connect our research with education and biodiversity conservation. And I also feel that despite all the environmental problems that we are currently facing, I feel that we still have many reasons for hope. I feel that there's a lot of goodwill out there. Many people are already concerned about the environmental issues, or searching for solutions and really doing great things. And so despite the bad scenario that we see out there, I really feel that things are starting to change. And for example, the millennium generation is very socially and environmentally responsible. So 80% of millennials will trust a company more if the company is socially and environmentally responsible. 67% will recommend products and services if the company is socially and environmentally responsible. 89% are likely to switch brands if the second brand is associated with a good cause. And 60% of millennials are likely to boycott a company based on its values. So I feel that companies are really starting to realize that environmentally friendly practices are actually key to their own survival, and they're starting to change the way they do things. So, for example, we have seen several success stories. So, for example, in the Brazilian Amazon, we have seen that the highest yield of farmers came on years in which there was lower deforestation. This increased rainfall and led to higher yield. We also see success stories from sustainable coffee farming. And we also see that some of the states in Brazil, like the state of São Paulo, which is the state that invests more money in science and research, this also turns out to be the richest state, which makes a clear connection between the importance of scientific findings. We are also starting to see pressure from the government, and the idea of raising taxes on firms that harm nature is becoming more and more popular. And we are also seeing citizens that are also starting to pressure their governments for increased environmental awareness. So for example, 886 Dutch citizens sued their government back in 2015. And as a result, the Dutch court actually ordered the state to reduce emissions by 25% within 5 years in order to protect its citizens from climate change. So this initiative started a precedent that could really be followed by many different countries. I also feel the Paris Agreement is another big reason for hope. So under this agreement, we have 185 countries that agreed to contribute to decreasing global warming. And even though we obviously need to think globally, it's also very important that we all act locally. And even if our individual actions seem small, I feel that these actions really, really matter. And so actually at every single moment, we are making lifestyle choices that are really the basis for, could really be the basis of a green economy and for a sustainable future. So we really have to be very aware and make good decisions on our daily basis. And of course, science alone won't save the earth, but in fact, no single sector has all the answers, which means that we really need to interact with people from different sectors and work together with different sectors so we can really find meaningful answers. And it's only through the collaboration across different sectors and disciplines that we will really be able to achieve a green economy and really be able to save biodiversity and the environment. So I just want to thank all of our sponsors. I also want to thank all of my lab members. I also want to thank all of the members of our Amazon project. And I also want to thank my family, who has really been supporting me a lot. And even though I'm always gone on trips, and who over the years, we all share very similar values, and over the years we are really together on this mission. So I want to close with actually a quote from my daughter, in which she says, "I have a dream that the trees will stop being cut down, and that the people will respect animal habitats, and that the ocean will be clean." And with that, I'd like to close, and I'm very much looking forward to our questions and Q&A session later this week. Thank you so much.
Oi, Lu!
Oi!
Maravilhoso!
Ai, meu Deus!
Foi
O
ótimo!
Ai!
Foi muito bom! Tadinha, tá cansada!
Para o recording, peraí.
Não, pode deixar. Eu vou